This is the Convergent Science Network podcast. Leading researchers in the domain of neuroscience, brain theory, and technology are interviewed by Paul Verschure and Tony Prescott.
All right. This is Paul Verschure with the Convergent Science Network podcast, together with my colleague Tony Prescott. And we're here with Matthew Diamond, who was speaking at our BCBT Summer School 2015 edition. Matthew, you were talking about the whisker system in rats, but in some sense you give your analysis of the whisker system an expansion, if you want, into a domain that is in some sense counterintuitive when you look at people who do work on rodents, because we start to really look at pretty complicated decision-making tasks. So how did you actually get from the study of the whisker system and its intricacies into this domain of decision-making?
Well, it took a number of steps, and I should say that one of my colleagues in this field always tells me that we do behaviors that are far too complicated. Why do we do such difficult? Why do we train rats in such difficult tasks? We would do better with a go, no go, whatever. And there's some truth to that. But we got there because we're in a cognitive neuroscience department, and we've always wanted to study cognition, that is thinking, decision making, perception, all these things together. And when I started in neuroscience, my PhD and as a postdoc, there were issues that we wanted to study. People knew a long time ago in the 1990s, people knew what were the interesting properties of the brain, some of the most fascinating properties of the brain, the intriguing things like decision-making and perception, but there just weren't ways to do it. And so all of us would say to each other and to ourselves, what I really want to understand is perception and decision-making, but I don't have the methods, they aren't available. So let's anesthetize the rat and give a controlled stimulus and measure the response to the stimulus. But it was always a compromise. And then in the last 10 years, there's been a lot of progress in instrumentation, in neurophysiological methods, in understanding of how to train rats that has allowed us actually to begin to explore the things that we knew for a long time we wanted to do, but we just couldn't get at them before. It's interesting that the training of animals is actually, some of it is a invention of the wheel, because a long time ago in experimental psychology, people did very, very interesting things with animals, going back to 1920s, 30s, 40s, and then neuroscience became much more fascinated with neurophysiology. Behavioral training was sort of put on the back burner and forgotten. And then in the last 10 years, there's been a rebirth of the attempts to study interesting behaviors in rats.
Right. But now for the whisker system, you made this distinction between, let's say, active sensing and receptive sensing. Mm-hmm. So why do you think that's an important distinction to make?
Well, I joined Tony years ago, 5-10 years ago, in projects in which we focused on what we called active sensing. Ehud Aysar was also very important in this way of thinking, and because the whisker system goes out to explore the world. It doesn't wait for things to happen. It makes things happen by a very deeply ingrained interaction between sensory systems and motor systems. And I bought into that and used that terminology. But when one is faced with active sensing, which usually means whisking, moving the head, moving the whiskers, and then we say, well, what if the animal's not moving the whiskers? What kind of sensing is that? And the natural terminology would be passive, because it's contrary to active sensing. So if the animal is palpating a surface or moving around an arena and feeling the walls, and we say that's active sensing, then when it receives a vibration without movement, one would be left calling that passive sensing. And I didn't think that was right. My students and postdocs and all of us in the laboratory observing the animals did not feel that they were in a passive state. We felt that they were actually actively controlling the whiskers in such a way as to collect a vibration from an external object. And so we refer to that now as receptive sensing. And when the animal creates the stimulus by its own movement, we call that generative sensing. And we believe that both are active. Mm-hmm. So there are two states of the system within the realm of active sensing.
So there's still passive sensing as another possibility here, where there's some unexpected stimulus on the whisker.
Yes.
But now, from the perspective of the whisker system and how you use it, mm-hmm. aren't you always operating in a mixed mode? And isn't there, to make that clean distinction, isn't that only holding in the laboratory, and it will be more difficult to maintain that under real world conditions?
Well, we just don't know enough about real world rats. We don't know enough about their natural history, what they do, how they do it, how they live, how they move. It's just, I hope that new research programs will begin on that. But as of now, we just don't know what they really do with their sensory systems. I think that there may be, as Tony said, something that is passive sensing, which means that the animal doesn't know what to expect and receives the stimulus that happens without being able to prepare the system for what's going to happen, because they don't know. So, for instance, in unexpected arrival of a predator, an unexpected stimulus could be processed by a passive sensory system simply because the rat is not able to actively set the state of processing.
Mm-hmm. So then, so let's assume we have the sensor system worked out. These rats are really great in using their whisker system for different kinds of discriminations. But now you're gonna use this modality in actually a pretty intricate task, right? Where also you described a number of stages that you want to manipulate, which starts controlling, let's say, the attention of the animal to the upcoming stimulus. Then there will be a first stimulus, this is a vibration on the whiskers that must be encoded. Then there will be a pause. So now we are dealing with the working memory task. Second stimulus. Then there's a comparison, a subsequent delay before the action can be executed. Then we get the go signal. The animal can choose left or right to get a reward.
Yes.
Okay. So if you just would look at this protocol and you would say, look, I'm gonna train my rats to do this, I think most people would say, look, Matthew, you're mad. This is just too complex. So how much time does it take to get a rat to be really sort of sufficiently capable of performing such a task?
Well, there are different questions that you can ask the animal to do in the context of this task. But I'd say that for the first level questions, you can get answers with about 2 months of training. If you want to introduce new variations or new variables, it can take as long as 3 months. And it varies from rat to rat. Some are very clever and learn very quickly, and some are slower. And we tend to be patient with the slow rats and try to train them till the end rather than discarding them. So we see differences between rats. But I think it's worthwhile for a number of reasons. First of all, just the fact that the rat can do this is informative, because, as I mentioned during the talk, this is, compared to the primate brain, it's a very small brain. It has many fewer modules in the cerebral cortex. So the question is, having available a smaller brain and a simpler brain in some way, are there things that the rat simply can't do? And what can it do? And so we're sorting this out. There are a number of laboratories that have progressively in the last 5 to 10 years introduced into the world of rats a series of primate tasks. In fact, there was an article in Nature about this 3 or 4 years ago. I think they called them the Rat Pack. Mm-hmm. And so there was the belief in the field of systems neuroscience, cognitive neuroscience, that rats simply couldn't do them. But it turns out that it takes a lot of training of the research team in order to learn how to train the rats. So we train ourselves, and we learn how to train the rats. And it's a question of finding the right methods, the right apparatus. And once that's done, the rats can do very primate-like things, even in the visual system. We all know that primate visual processing is extremely advanced, and we're not saying that rats can have visual perception up to the level of primates, but there are some properties of visual processing that 5 years ago people didn't believe that rats have, and they do. Turns out they do have them. I'm referring to invariance according to position, according to size, according to viewing angle. My colleague at CISA, Davide Zoccolan, has found that rats do have these invariance capacities, and people thought they were a property of primates. So if you put the time and effort into finding the right kind of stimuli, the right kind of training apparatus, the right training regime, rats can do primate-like things, and this alone tells us something.
Mm-hmm. And is this really to use rats as a substitute for primates in brain research, or is there some other reason to do this?
Well, I think that's one of the outcomes is to be able to do certain kinds of cognitive neuroscience, perceptual neuroscience in rats, and not have to rely on primates. That's certainly one benefit of this, but it's not the only reason for doing it. Even if there were no constraint on using primates, it's interesting to know what rats can do because they do it with a very different organization of the brain. And the fact that a complex task, a primate-like task, can be done with a very different brain structure tells us something about the way the brain works. It tells us that the brain, in particular the cerebral cortex, can find solutions to doing certain kinds of computations, but in a different way. And discovering how the rat brain can accomplish this actually tells us a lot about overall brain organization.
So I'm a bit surprised you say that, because isn't part of the argument you'd want to make is that though the rat brain is simpler than the primate brain, there's some fundamental mechanisms in task-like decision-making which are probably similar, and the neural substrates may even have very interesting similarities.
Yes, they may, and the point is these mechanisms can be installed in a circuit with fewer neurons and with fewer modules, and yet somehow the same computation can be accomplished. For instance, in what I talked about today, there's a very clear working memory component, and if you use the word working memory with systems neuroscientists, they immediately think of prefrontal cortex. Now, rats may have a prefrontal cortex. They probably do, but nobody's absolutely certain what region it is. It's very hard to find an analogy or homology between rat prefrontal cortex and primate prefrontal cortex. Some people believe that the rat homology to prefrontal cortex is a prelimbic area along the medial wall of the cortex, but nobody's really sure. And yet, with this possessing or maybe not possessing a prefrontal cortex, rats can carry out working memory as well as primates, nearly as well as primates, and so they're somehow getting these computations done with a different brain structure, and it's interesting to know how they do this.
But I think your data, but maybe we should go through that first. But I think the physiology that you have gathered on this task is actually also partially answering that question, whether it is that different or whether there are similarities. Mm-hmm. Right? But maybe we can come back to that once we understood the task a little bit better. Because so what now happens is that the animal is exposed to two stimuli. These stimuli are a certain velocity of movement of this plate that you manipulate, and the variance of that velocity you control. So
Yes.
That means if you want the overall amplitude of the signal you're integrating is now controlled, is different, right? But the second parameter you control of your stimulus is now the duration of the stimulus.
Yes.
And the question now is, okay, to what extent can the animal make this discrimination and say, oh, this was shorter or longer, like stimulus 2 was shorter or longer than stimulus 1, and this informs me whether I should go left or right to get my reward. And the reward is a liquid reward, and animals are water deprived, or it's...
Yes, it's actually fruit juice.
Okay, so they're not food or water deprived.
They have they're water restricted. They have they drink a lot during the task, because in a few hundred trials they get almost as much liquid as a rat in its home cage would have in the course of a day. Then after the training session, they also have another hour to top up if they feel like it, and then they're restricted until the next session. So they arrive in the session thirsty, but certainly not in any sense in physiological stress.
Right. So then, so now we have this parametric control of our stimulus, and you made the point that this actually is a qualitatively different task than when we have to do some sort of categorical decision making. Let's say I show you Tony, and then I show you a car, and then you have to go left or right, right?
Yes.
So what is the principal difference in your mind, certainly from the perspective of the rat brain, between that categorical decision making and this more parametric decision making?
Okay. In the parametric decision making, we actually presented two kinds of behavioral paradigms. One is in which the rat or the human subjects, we also study humans, has to compare this intensity value that you talked about, this variance. And in that case, they should measure the physical characteristics of the stimulus, but optimally, they should not attend to the duration of the stimulus. Then as a second task, we ask animals and human subjects to judge the duration of the stimulus while not attending to the intensity value. So in both these cases, the comparison is what is, as you say, a parametric one, which means that every stimulus is distributed along the same dimension and vary only in their location along that dimension. So we can draw an axis, which we could call variance of the stimulus or intensity or amplitude. We could use different terms, but we can draw an axis, and then every stimulus simply has one position along this axis. And the object of the brain then is to find the relative position of two stimuli to compare them. And this is parametric because there's one parameter that defines a stimulus. In the case of categorical stimulus comparison, which ecologically is hugely important, but it's a different kind of strategy for the experimentalist, because the stimuli that have to be compared, let's say, living thing in a visual system, in a visual task, you could ask the subject, do you see a living object or not see a living object? And you could show an image for a half second. That's an example of a categorical task. The stimuli that have to be processed may engage different sets of neurons. So the comparison could be between one stimulus that evokes activity in one set of neurons, a second stimulus between neuron that evokes activity in a second set of neurons, and eventually the brain, of course, has to converge the two populations in order to make the comparison. But at this stage of our understanding, we don't know in categorical tasks which we can't identify exactly the sets of neurons, and we don't know where they converge. And so we selected a task in which we could actually study the coding, the computations done by neurons, in that the exact same set of neurons encodes both stimuli.
Yeah, because the difference now is, of course, that the neurons that we're going to look at are in some sense forming an analog representation of the stimulus itself.
Yes.
Well, in the categorical case, that would not be the case. Well, a categorical representation still has to be built on a preceding parametrically controlled one.
Yes.
So is it possible that you are then looking at a lower, let's say, processing step in the hierarchy than what would play out when we have categorical decision making? Or you think that in this part of the rat brain where you're looking, that you would also find categorical representations if you knew what to look for?
So we don't know. I think rats can make, for sure, they can make categorical decisions. For example, there's very systematic studies in the olfactory system where rats compare odors. These I would consider to be categorical because the odors are different from each other. They engage different receptors. They create activity in different populations of neurons. And then at some stage, perhaps in the olfactory cortex, we're not sure where, there's convergence and a decision is made. So rats can do this. We've explored in rats, and we've had some success. We've also done a large set of studies in humans that I didn't present today, in which the comparison is actually made between modalities. So we can have an acoustic stimulus that has some value along a dimension, an intensity dimension, if you wish, and a tactile stimulus that has another value along a dimension, and then the subject has to compare the two stimuli in different modalities. So the first thing they have to do in order to do this task successfully is create some scale whereupon stimuli in different modalities can both be projected. So you have to, for instance, if you want to, you could call the scale from 1 to 10. So the subject has to learn to put an auditory stimulus along the scale of 1 to 10, tactile stimulus along the scale of 1 to 10, and then make a comparison. Humans can do this with a lot of work. We got some rats to do this, but it's very difficult for them, and the rats actually had good days and bad days, depending on how much effort they wanted to put into it. So it's very hard for rats. It's quite a challenge for humans, but humans can do it. So that actually requires, to do the task requires a convergence between the auditory system and the tactile system, and human subjects can do this.
Right. So now, if we look at the stimuli you're using, so these parametric stimuli, what is, let's say, the discrimination threshold at which, and also the just noticeable difference at which rats operate? So how big a difference can they really distinguish between these different stimuli?
Well, that's a great question. I don't recall that we've actually I don't recall that we've actually looked at our stimuli. We haven't quantified it exactly in terms of just noticeable difference. I think that we could do that computation. We haven't, but I think it would require examining the psychometric curve and then making a model for how far along the psychometric curve, how different two stimuli would have to be to produce a different, a measurably different choice in the animal. And we have the data available, but we have not made exactly that calculation.
Okay. So now, if we talk about the psychometric curve that you defined or extracted from the performance of both rats and humans, they're sort of similar, but certainly not identical as far as I got your data. So what are the important differences here between the psychometric curve that the rats maps the properties of the stimulus to rat performance versus those you find for humans?
Well, as you saw, the rats can perform the task. The average, if we take a set of rats and a set of humans, the average value for the human is better than the average value for the rats, measured in either as percent correct or as a more derivative measurement is the slope of the psychometric curve. And these are two standard measures of performance. On average, rats are inferior to humans, but there's individual variability in both species, in humans and rats. And this is one of the many, many interesting, funny things that we find in rat behavior and rat psychophysics. If you talk to a typical psychologist, human psychologist, their impression would be that humans differ very much from each other, but a laboratory animal like a rat, they're all the same. It turns out that rats vary among the individual variability is as much or more than that in humans. So a good rat in our data set a rat that performs really well is actually better than some of the humans that perform less well. So on average, the humans are better, but there's very significant overlap in the two clouds of performance.
Wouldn't you expect the rats to have less variability because there's less general genetic variability in the population of lab rats?
That would be our expectation. Our rats are sort of outbred, so there is quite a bit of variability. And we have completely different behavioral procedures where we confirm a lot of variability between rats, but they're not completely inbred.
But now in some sense we always make these normative judgments, right? We see these psychometric curves, which means we're making assumptions about optimality. And it's not unreasonable to assume that rat brains have been optimized to satisfy somewhat different constraints than human brains. So in that sense, the biases and the priors in that rat brain might already be different from the human brain, and maybe given those priors, the rat is behaving optimally.
Yes.
So what do you see as the most significant differences in the biases that the human brings to bear to the task and that the rat brings to bear on the task?
Yeah. Well, the rate of learning is of course radically different. In the case of human subjects, we have a training session in which they are presented with a stimuli in the task and they make a choice, and the computer tells them if they are correct or incorrect. We don't give them a verbal instruction to tell them what to extract from the data. We don't tell them this is what you're going to feel. This is a parameter of the stimulus that you have to extract, and then you have to compare this parameter in the first to the second. We don't do that because we think it would create lots of biases and lots of top-down processes and too much thinking. So instead, we simply let them feel the stimuli. They receive the stimuli. They make a choice, and the computer says correct or incorrect. So during the training session, the human subjects are in fact testing possible hypotheses about what the rule might be, but we don't tell them the rule. Then trying various rules, we're not sure exactly what the mental rules that they test are, but trying various ones, they eventually arrive at a choice based on the thing that the computer actually employs as the rule, and they start getting it right. Mm-hmm. And so when we see them get about 80% right in blocks of 10, then we judge them as having figured out the task and the training is over and they can move to testing. So in humans, this can take 10, 20, 30 minutes. In rats, the training takes, as we said before, 1 or 2 months. They have to go from through multiple stages of training, beginning with simply letting them explore the box. Then they have to learn that they have to go into a specific sector of the box and put their nose in a specific place. We can't tell them to do that the way we can tell human subjects to hold their finger here. We just have to train the rats to do that, and each of these takes a week or two. So when you get through all the stages, 2 months have passed. So that's a major difference in the training. In the performance, one difference you noticed is that on very easy stimuli that we know that the sensory system can process correctly, humans can perform at 95% or even 100%. Rats, even for very easy stimuli, will make the wrong choice 10 or 15% of the time. And this is something that psychologists sometimes call lapse rate, which means errors that are not due to sensory processing, because we think that the sensory system really can handle those trials. So the lapse rate is something that distinguishes rats from humans, and there are many theories about why rats make lapses. One of the most interesting theories is that when they make this kind of error, what they're actually doing is making sure that the rule has not changed. That is, if they go to the opposite side to where they know they'll get the reward, that if they go the opposite side, they want to confirm that there is in fact not a reward there.
So it means rats might be more tuned to exploring the task space and to keep on exploring the task space because they might have evolved for environments where the contingencies are less stable as humans have.
Absolutely. I think humans, if they discover that there's a rule, then they are willing to act according to this rule until they have evidence that it doesn't work. Whereas rats are much more prone to exploring the environment because perhaps they've evolved to believe that environments can change, that rules do change, and they want to make sure that something hasn't changed.
Do you also believe that based on that rats would operate at a different level of, let's say, anxiety and certainty than humans?
Possibly. I mean, they're certainly, they may be more anxious on the basis that they're working for their daily water. They get as much as they need. If they don't get enough liquid during the training session, the testing session, then they can drink for a couple of hours afterwards. So they're not deprived of water in any way, but they know it's something that's important to them, and they want to get it right.
Mm-hmm.
Surprisingly, humans also want to get it right, but for different reasons.
They don't want to disappoint you, I think.
Yes, and they're most of the subjects are students, and I think that they want to be better than the other students. I think there's some rivalry.
Right. So then, so here we have these psychometric curves now for rats and humans. We know rats are slightly worse than humans, but not dramatically so. The overall psychometric curve is a bit flatter than in the human case. So also the extremes are a bit more compressed, as far as I could tell.
Yes.
That basically means we know now the rat can perform the task.
Yes.
So now we want to know, okay, how do then these two properties of the stimulus that you're manipulating, which is intensity and duration, affect task performance? Right. So what can you say about that relationship?
Well, the parameter that we first explored was the perception of the value of intensity, the parameter of intensity. But we arranged the experiment in such a way as that intensity is not a value that emerges instantaneously from the stimulus. It's a stochastic stimulus. It's a vibration, but a vibration which is created by sampling a normal distribution. So you could call it modulated noise in a way. So the effect of this is that at any given instant, there's not really a complete signal available to the sensory system in order to make a choice. Instead, the information necessary to make a choice, to make the judgment, accumulates over time. That's the nature of a stochastic stimulus. A stochastic stimulus is defined by probabilities, by a statistical distribution, and with an increasing number of samples, the statistical structure becomes more evident to the brain, to whoever is sampling the stimuli. So from this, our prediction is that if the brain is actually accumulating evidence and using the history of events during the stimulus, then the performance would be better with a longer duration. And this is what we found in both in rats and in humans.
Mm-hmm. But
you said that there was some evidence that maybe rats wouldn't be good at accumulation in that way.
Yes, there have been other perceptual tasks that have been explored in rats, change detection kinds of tasks, in which there's been evidence that under some conditions, rats do not use a long history of tactile stimulation in order to make the choice, but use only very recent history. And apparently, this depends on the way that the animal, what it decides to optimize in the task.
So is it possible that your results are consistent with those other findings because your task emphasizes you can't solve your task without evidence accumulation?
Yes, it's true that the task was actually set up in such a way as to reward evidence accumulation. It was set up with a statistical structure such that performance is better if evidence is accumulated. But that doesn't necessarily impose on an organism to do it. An organism can do the task without evidence accumulation. It would simply have not good performance.
But then what you showed, which was very surprising, is that this integration process of the information, the evidence provided to the animal, is following a summation rule and not so much an integration process like a multiplication. And this had to do with an interaction effect between intensity and the duration of the stimulus.
Yes.
So how did that exactly play out?
Well, the task involves comparison of two stimuli, and the two parameters that we can control are the intensity value, which was the variance in the vibration. The higher variance is perceived as being more intense, and the duration of the stimulus. Of course, there are many variables in the experiment that we didn't talk about today. For instance, the delay between the first and second stimulus is extremely interesting because a longer delay puts a heavier load on working memory. Today, instead, we focused on the parameters of intensity and duration. And our first question was whether performance in humans and in rats would vary according to the duration of the stimulus. And as we were saying, the statistical structure is such that an ideal observer, a machine doing the task with all available data, would in fact do better with a longer stimulus. And we found that is the case under the conditions in which the first and second stimulus were of equal duration. Then we then varied the duration, but the duration but included trials in which the first and second stimulus were not of equal duration, which we call the unbalanced condition. And in the unbalanced condition, we found, to our surprise, that the subjects, both humans and rats, did not measure intensity as an average over time, but instead with some form of summation, as we saw, not a linear summation, but with some form of summation that caused a longer stimulus to be perceived as stronger. So it was not purely an averaging, but some form of summation.
Mm-hmm. Right. But do you see this now as, let's say, I have to estimate, you could say I can estimate these two parameters of duration and intensity. But now if I would integrate over these parameters separately, then duration might give me, let's say, a false sense of intensity. Because imagine I just have, let's say, a clock, and I'm just integrating from this clock longer, so my signal gets higher, and that might then start to have a crosstalk, adding noise, if you want, to my intensity estimate. So do you see it in those terms, like a crosstalk between informational channels? Or do you see it more as an intrinsic bias that the system has?
It's an intrinsic bias. It's not the longer stimulus adds noise. In fact, a longer stimulus reduces noise, in the sense that subjects perform better if the two stimuli to be compared last 600 milliseconds each, as opposed to if the two stimuli last 100 milliseconds each. So the longer stimulus doesn't add noise; it actually adds precision to the judgment of the statistical properties. The problem that I think the perceptual system has, which it is not able to overcome, is that it doesn't have an absolute clock. If the brain had an absolute clock, then it could measure the total activity occurring over a stimulus and then divide by this absolute value. But it's not available in the brain, and it's for some reason it's not there or it's not used, and so not knowing exactly how much to divide the accumulated value by, there's a bias such that the longer stimulus feels stronger.
And this effect you see as a linear effect. So if I double the time, then this subjective sense of intensity is also doubled, or it follows an exponential curve, as you showed sagrammatically. Or could you imagine that at the level of neural substrate, it might be, let's say, more discretized, or it might have different sensitivities because it has to write, for instance, on oscillatory activity?
Yeah, we don't know in exactly that level of detail. We built a model that simulates behavior correctly and accurately when the summation was done not linearly but exponentially, with a exponentially decreasing weighting function, so that the onset of the stimulus contributes to the perceived value with the highest weight, and then that weight decreases exponentially with a time constant of about 150 to 200 milliseconds. And then we compared that to the expected results if the weight of the stimulus increased over time, so that the end of the stimulus had more weight, and we found that the results were consistent with an exponentially decreasing weighting function, not increasing.
Mm-hmm. Right. But now so in some sense, you put now the process that would account for this bias at really the sensor processing end of the pipeline. While in between, we still have our working memory buffer. We have another integration stage that has to come to the decision.
Mm-hmm.
So why do you put the full burden of that bias at the perceptual end? It might also be, let's say, some sort of capacity issue in your working memory, for instance. Right?
So we don't know where this weighting function occurs, but we've seen that if we look at the activity of sensory cortex, primary sensory cortex, which in rats is for the whisker area, this is called barrel cortex. The activity of neurons there does not reflect a weighting function. It's so if we try to decode the choice of the rat based on the activity in sensory cortex, the choices will not reflect the duration of the stimuli. So the effect of the duration must occur after sensory cortex. Another region that we're looking at is premotor cortex, which is in front of sensory cortex, and there we find many neurons that have activity during the time between the first and second stimulus, where the activity has a very clear relation to the first stimulus. So at first glance, one interprets these as working memory neurons. Mm-hmm. Many laboratories have found neurons like these. The Roma laboratory in Mexico City has found many neurons like this in exploration of primate cortex, and the interpretation is that these are neurons involved in working memory. Now, when we look at how in rats the working memory neurons encode the first stimulus, we find that the firing rate is but has a stronger, better statistical correlation with the first stimulus if we consider the first stimulus as the perceived value of that stimulus rather than the actual physical property of the stimulus. In other words, we find that the activity of a neuron that varies according to first stimulus intensity will fire more if the first stimulus lasts longer, even if the intensity during that stimulus is constant. So as the stimulus continues over time, we know from a lot of behavioral work that as the stimulus continues over time, the perceived intensity increases. That this came out from the psychometric curves. At the same time, we find that neurons in premotor cortex, if the firing is positively correlated with the stimulus intensity, then those same neurons fire more when the stimulus lasts longer. So the firing rate actually reflects the stimulus duration. So the firing rate in premotor cortex reflects the perceived value of the stimulus rather than the average value of the stimulus.
Mm-hmm. Right.
Do you do a control where the intensity is not varying randomly within a given range, but is fixed, and so you get a continuous stimulus at that right target frequency?
So two stimuli are being compared, and
so rather than getting a noisy stimulus with an average intensity, you would just have the stimulus continuously at the target value. That would be an easier task.
Something like a sinusoid, for instance.
Yeah.
No, we have not used sinusoidal stimuli in this stage of the experiment. We did in some very preliminary pilot studies and found that rats had a hard time maintaining attention for pulse trains and for sinusoids. That is more difficult to convince the rat to remain in the nose poke, to remain immobile with its whiskers in contact with the plate. For the first stimulus, the interstimulus delay, and the second stimulus, to wait for the go cue when the stimuli were sinusoidal. They tended to abort the trial, that is, to leave early. So to put in layman's terms, periodic or regular or constant stimuli are not interesting.
Right.
And for some reason, we're not sure why, the noisy stimuli are interesting. The rat is very willing to wait for the entire stimulus. We can make the stimulus, if we want, even 1 second long, 2 seconds long, and they wait.
It's interesting. So, I mean, one thing that occurred to me is that, so you have a task in which the stimulus is varying along two parameters, intensity and duration. But success in the task requires that you just attend to intensity and
yes.
that you ignore duration.
Yes.
It's because it's put there as a kind of confound.
Yes.
And your model is assuming that they are more or less successful in attending to the intensity. But presumably, I mean, neither the rat nor the human knows that this rule is just about intensity. So they might be working on all sorts of theories about mixtures of intensity and duration. And if you give either duration or intensity on their own, then they can solve those problems too. So I'm just wondering, in the model space that you've explored, have you explored all the potential variants of rules that the animals could be using to try and maximize performance on this task? Are there rules where they might, for instance, you might do reasonably well by sort of taking the duration into account in some way?
Well, the computer determines we put a reward rule into the computer, and that's applied to the rat or to the human during the experiment. And the reward rule, if we do an intensity experiment, the reward rule is intensity. That's what has to be compared. Or we can, in different rats or different human subjects, we can change the rule and such that the subject is rewarded according to duration, and to perform perfectly should ignore intensity. Now, if we consider the first case when the rule is intensity, there are many, many trials in which the two stimuli have the same duration and vary only by intensity. So an example would be a 400 millisecond stimulus followed by 400 or 600, 600, 200, 200. In all of those cases, there's no choice could be made based on duration. In fact, the rats and the humans perform very well according to the intensity rule. So we have no reason to think that when they're actually doing the intensity task, that they might be doing a time task. They know that when they're doing intensity, they're doing intensity. The problem that they have is that when they try to accomplish the intensity task, the sensory system is confounded by the duration.
Well, that's your reading of what they're doing. I mean, is your assumption there that intensity is more salient to them than duration?
No, because we can train rats and we can train humans to do duration.
Yeah.
The question is the reward rule. In fact, there's actually there's no requirement that we use a different stimulus set. If we have two parameters to be varied, the intensity and the time duration, we can have stimulus pairs that differ in one or the other, and the subject is simply rewarded for making a choice based on one parameter and not the other, or the second parameter, not the first. So we don't even have to change the stimuli. We just change the rule, and we find that a subject trained according to one rule makes its choice based on that rule, and a subject trained on the other rule makes its choice based on that rule.
You still have some kind of interaction effect, because with the intensity, the longer stimuli are easier to classify. So this makes your task rather different from a lot of more standard judgment tasks of that kind.
Yeah. Right. So for the longer stimulus, there is more signal available for making the discrimination. And we found that, in fact, the discriminations are made better, both by humans and rats. And this confirms work done by the Roma laboratory with vibrations applied to the finger of monkeys, and also in visual system detecting the direction of noisy moving dots which move in a coherent or incoherent way. Performance is better for longer stimuli. So we've simply confirmed that for stimuli in which more information, more signal is available in a longer time, even to the ideal observer, there's simply more information available, that in fact rats and humans perform better. So that's a confirmation of many, many different studies. And the confound between time and intensity was something that emerged from our experiments. Then we went back and in literature and looked for this and found that there have been reports consistent with this.
And are you able to look at the sort of learning history and infer from that anything about perhaps the strategy, certainly in humans, so you could imagine you have a strategy of testing various hypotheses, but even in a rat you might imagine that there was some pattern of changes in the trials where at one stage they have no idea, and perhaps another stage they're testing that it's about intensity, these kinds of things. Is there anything to see from that?
Yes, during training, rats, of course, have very low performance as they're learning, but they make a choice, and they might very well have some rule in mind, some algorithm by which they're acting, and it's not working, or maybe by chance works 60 or 65% of the time, and they think that that's fine. So we have to simply train them day after day, and if they had in mind a rule that was not the one that the computer is set up to use, then sooner or later they'll discover that it's not reliably giving them the reward.
So now, so you made a model to explain the performance, or the mapping of the stimulus properties to the performance, and in there you see that you have this very fixed time constant at which you are ramping the sensory evidence that comes in.
Yes.
And you just mentioned 150 milliseconds or something like that. Do you see this really as an invariant that is sort of wired into the human and the rat brain, or this is also in turn dependent on task properties?
That's a really interesting question. We've looked in. We've been able to, from the simulation, the model, we've been able to extract this time constant tau for a number of rats, a number of humans, and to our surprise, and I think probably it would be surprising to many colleagues, the tau, the time constant, was actually slightly longer in rats than in humans, about in the order of 150 to 175 milliseconds for rats and about 100 to 150 milliseconds for humans. So while many of us would have predicted that humans accumulate evidence over a longer history, it turns out that rats accumulate evidence over a longer history. That was a big surprise to us. Under the same conditions. Now the question is whether this tau is something intrinsic to the brain. Might it be the same for different kinds of stimuli, maybe even for different modalities? And we don't know, but our guess, we would still have to do the tests to check this, but our guess is that tau does depend on the stimulus conditions. From a mathematical point of view, if the stimulus is stochastic, it has some frequency properties, what we can think of as a correlation time. That is how much time has to pass for the stimulus to be uncorrelated with what it was in the past. How much time has to pass before the stimulus becomes completely unpredictable, completely independent. And in the conditions we've used, with a filter of about 150 milliseconds, the correlation time is in the order of 10 milliseconds, plus or minus a few milliseconds. And so that means that in 150 milliseconds, the rat could sample, the sensory system could sample approximately 10 to 20, roughly 10 to 20 independent samples. If the correlation time were longer, that is, if it took the stimulus longer to achieve a value unrelated to the previous value, for example, making the filter a lower pass, then it would take longer in order to accumulate the same number of independent samples. Therefore, to achieve the same performance, the sensory system would have to actually sample for longer. So one would expect that tau may adapt to that by increasing the integration time. This is a bit mathematically complex, but I hope it comes through.
But then, so the point would be that either have some sort of attenuation factor of the evidence I accumulate, or I might be sitting in some oscillatory dynamic that is basically dictating to me that, okay, the early samples will have a higher impact on the information integrate than later samples. So how do you see it more as a continuous attenuation factor, or do you see it more as sort of an oscillatory sampling process?
So until we have evidence to the contrary, our guess is that this is a continuous weighting function that changes continuously over time. We haven't really looked for and therefore haven't seen any signs that it might be oscillatory, maybe at a gamma frequency or something like that could have some role, but we simply haven't looked at that.
But then it is a weighing function that in turn might be task dependent. So that means for different tasks and different, let's say, stages of learning, the weighing function will change. Yeah. Right?
No, for sure it does. I mean, let me try to pull out an example that might make sense intuitively, although I'm not working through this mathematically. But if I ask you, is the climate of the Earth changing? You would collect samples. You'd go back in history and use ice core or whatever might be available and collect samples over a long time to find out if the climate of the Earth is changing. So the period of time over which you'd have to collect samples in order to give us an answer would be many hundreds or thousands of years. If I ask you, is something that changes in the order of seconds, you would need many fewer samples. So clearly in any kind of task, in order to make an estimate, in order to estimate the properties of some time series of data, you will need a different number of samples in order to make the estimate.
So from the model, this now follow logically, right? You also predicted that if you would compare now a primacy or a recency effect, like more evidence in the beginning or in the end, that actually for both the rats and the humans, you would see primacy, and that's also what you observed.
Yes.
Right. So this was actually a really nice prediction that came out of the model. But then the next step is, now that you have the model in your hands, okay, what are the neurons really doing? Okay. So with that, you went to a frontal area in the rat brain, and you start to look now at the neural responses in this task. So what were the features of the neural responses that stood out for you in these populations of neurons that you measured from?
So the data that I presented today came from two regions of the cerebral cortex. The primary sensory cortex, which, as we said, in rats, it's called barrel cortex, the cortex that receives input from the whiskers. And then we looked at a more frontal region, which is usually called premotor cortex or whisker motor cortex, or it's also being called by Carlos Brody frontal orienting field, FOF. So different people have different names for it. So I showed some evidence from that area as well. In the sensory cortex, we found that around half the neurons have a very clear relationship in their firing to the stimulus properties, but what the neurons report in their firing is only the most recent events of the stimulus, the last 10 or 20 milliseconds. So these are what we call local coding neurons. They encode what happened just immediately, just a few milliseconds ago. What happened 100 milliseconds ago or 200 milliseconds ago has very little impact on the likelihood of a spike at any given time. So at time t equals 0, the stimulus value at t minus 200 or minus 250 has no impact. But what happened at t minus 10 or 15 milliseconds has a large impact. So the neurons report the most instantaneous, most recent value of the vibration. So this is an essential element for the brain to be able to reconstruct the stochastic stimulus, this noisy vibration. The noisy vibration is made up of a sequence of local events, but any given local event does not define the stimulus. And so there has to be integration done after the sensory cortex in order for the brain to appreciate the overall statistical structure of the neuron, of the stimulus, sorry. So the sensory cortex neurons are local coders, and the integration has to occur elsewhere. In the frontal region that we refer to as premotor cortex, the activity of neurons did in fact reflect the overall statistical structure of the stimulus, but not the local history. So, for instance, from looking at the firing of a neuron in prefrontal cortex, we cannot say what happened in the whisker vibration 10 milliseconds ago or 15 milliseconds ago, but we can say what has happened over the course of the last 100 or 200 milliseconds. Mm-hmm. So those neurons already reflect integration, but we're not sure if they're doing the integration or are receiving a neuronal signal that's already processed.
Right. But now, would you see these primary sensory neurons as a wavelets, which is like neural wavelets that are now encoding a time series?
Yeah, that's one way to look at it. In fact, our colleague Rodrigo Quiroga is looking at spike trains as through wavelet analysis, and it's actually quite a promising approach.
Mm-hmm. Right. So now we have the prefrontal or the premotor area neurons looking at the signal at a longer time window.
Yes.
But can you correlate the neural response directly with the parametric control of your stimulus, like duration and the width of the vibration distribution?
Yeah, the neurons in the premotor cortex have most of them, those that do have firing that's related to the stimulus, are better correlated with what we would call the perceived value of the stimulus than the physical value of the stimulus. That is, we have each stimulus is characterized by the intensity, which we call sigma, and by the duration. The neurons in premotor cortex are, when they encode the memory of the stimulus or they encode the choice made by the animal, their activity is better related to the value of sigma after we take into account the effect of time.
And that's one set of the PFC or the frontal neurons. And then there are others that encode the working memory element, or is that what you're talking about, the working element?
So we've seen in premotor cortex as Ranulfo Romo, who is present today, he noted that he's seen the same thing in primate premotor cortex, that there's really a mixed soup of neurons there. It's incorrect in our data and his data. It's incorrect to believe that one module, one region of cortex is working in a homogeneous way. One sees in the same trainings, in the same test session, even recorded adjacent electrodes, in other words, neurons sitting side by side, do very different things. In premotor cortex, we see a mixture of neurons that include those that encode the stimulus as the stimulus occurs, but not the local history of the neuron, but rather the time integrated value of the neuron during the time integrated value of the stimulus, of course, during the stimulus presentation. So those are online as the stimulus occurs. Those neurons encode that stimulus. Other neurons encode the preceding stimulus during the delay interval between the two stimuli. So those neurons seem to participate in working memory. And then still other neurons encode during the second stimulus the value of that stimulus, the parameter of that stimulus, and others the choice that the rat has to make based on the comparison between the first and the second stimulus. And to make matters even more complicated, some neurons encode combinations of these different features. That is, it's not unusual for a neuron to encode the stimulus as it occurs and the memory of the stimulus, whereas another neuron may encode the memory but not the stimulus as it occurs. So there's really no single label that you can give to the full population of neurons.
So you can see why there might need to be different neuron types in order to solve this problem. But if there's no topography, as you suggest, then we have a real challenge for reading out what is the right answer here. So it's not just a case of looking at average firing in, say, the comparison neurons, because they're intermixed with the working memory neurons, and so on. So what do you think have you got any idea of how that readout might happen?
Well, I think that there's two issues, I would say two ways of looking at this problem. One is the computation that's being done physiologically by the cortex, and the other is the readout. So about the computation, I would say we're close to the starting point. We just don't know what it is. We don't know how, for instance, how a memory is stored for 1 or 2 seconds. We just don't know. We don't know whether firing rate is, which is what we've looked at, and most laboratories look at, we don't know if firing rate is actually the memory or if there's something beyond firing rate. There could be population codes, there could be trajectories through multi-dimensional population space, there could be latent synaptic weights during the delay interval. There are many ways, many theories for how memory is stored, and we really can't confirm or exclude any of these. So about this, we really don't know. We don't know much, and we don't know the transformation from the feeling of an ongoing stimulus to a memory of the feeling. This is completely unknown to us. The second point that you raised, decoding, well, we can quite easily make networks that take our activity and decode it, and that's not difficult to do. We can, from the same population of neurons, we can look at the population from two different angles, and by one angle we see a memory, and from another angle we see a choice. This has been shown, for instance, in a paper recently. It's the author Valerio Monte, I think, if I'm not mistaken, is the first author of the paper, from neurons in primate prefrontal cortex, where there were two features of a stimulus, I believe color and motion. And the monkeys were trained to do either a color task or a motion task. And he found that the same neurons encoded both features and could be decoded from the activity of those neurons. Either the color or the motion could be decoded. So it's not that difficult using sort of offspring of principal component kinds of analyses to find dimensions whereby a certain property can be decoded. We can do that, but what we don't know is what makes the neurons fire that way.
So from the population response, you can decompose it into different components, yes. One of which gives you a good indication of the animal's behavior, its actual response.
Yes.
And so we're assuming some downstream system is able to do that decomposition.
Yes.
But now the standard model of decision-making are drift diffusion models where we just integrate over firing rates. And in some sense, if you look at your model or your physiology, your data, then you also see some very marked or task-specific modulation of firing rates. So is the minimum model that we could apply to this then exactly that, the drift diffusion model?
Yeah, this has something in common with drift diffusion, and it's the kind of stimulus statistically that has been explored for drift diffusion. I think that one of the main differences is the behavioral arrangement in this task does not ask the rat or the human observer to reach some threshold and make a choice. And we don't give them the option of collecting as much evidence as they want and then taking the action. If in those cases, it's been argued that the animal or the human collects evidence until they drift to a threshold, and then they make the choice. In our case, we determine the stimulus duration, and the animal and the rat and the human are required to wait for the entire stimulus. So that may take them beyond the threshold, or it may not. They may not be allowed to reach the threshold. That's determined by the stimulus duration.
Okay, but that would still mean that the decision variable could also be reflected just in the firing rate, and then it's a matter of reading out that firing rate. Yes. It's not necessarily my favorite model, but when you have market sort of correlation, if you want, between the performance and the firing of these neurons.
Yes.
Of course, these are good examples, right? But in all the physiology you have seen, in the end, if we're close to the decision moment, you can actually predict from the firing rate of the neurons where we go left or right.
Yeah, and we've also looked at error trials, and on error trials, those neurons which have on correct trials a correlate in their neuronal firing rate to the choice of the animal have that same correlate on error trials. In other words, if a neuron fires at a high rate when the rat is going to turn right on correct trials, and a low rate when he turns left on correct trials, then on error trials, the neuron will fire for turning right and not fire for turning left, even though it's an error. So many of the premotor neurons that are choice selective are choice selective whether the choice is correct or incorrect.
Right. So this is really amazing, right? Because we're now going from a signal-dependent response in the nervous system to, let's say, a subjective state-dependent response. And with that, actually, you're really close to explaining this task. And then at the end of your talk, you also showed how actually it can have some diagnostic value if we look at humans, which was a very surprising result where you showed that, well, actually, in performing another variation of this task, humans that are at risk of schizophrenia actually show a very different kind of performance.
Yes.
So what is the salient feature there in your mind?
So we have just a few subjects who are completely healthy, yet the family history suggests that they're in the category of what's called at risk for schizophrenia, meaning that there's something in the family genome that may give a predisposition. Yet at the time of testing, they're absolutely healthy and symptom free. So the task that we gave to both to the healthy risk-free subject or low-risk subjects and the healthy at-risk subjects was a time duration comparison. So they received 2 vibrations sequentially and had to judge whether the first or second duration was greater. And among the confounding factor was the intensity. So this is motivated by the fact that the previous work had found that judgment of intensity was confounded by duration. We wondered whether perception of duration is confounded by intensity. And we found that in both groups of subjects, in fact, the perceived duration was confounded by intensity, in the sense that the stronger stimulus, the higher variance, the higher intensity stimulus was on average perceived as with a bias towards a longer duration. So to put it in very short and in rhyming words, longer feels stronger. Shorter duration feels weaker. And that this is the interpretation of the psychometric curves. So subjects were not able to purely exclude the intensity of the stimulus from the duration, even though the task would require that for perfect performance. in the subjects that were healthy but might be at risk due to family history, the effective intensity was much more pronounced. In other words, they were less able to exclude the irrelevant stimulus feature from the computation. It's to do the task perfectly, the brain should discard the intensity information, not use it in the computation, and do the computation based purely on time. That was what the rule was in the task, is measure the time. And these subjects showed a much stronger effective intensity. So essentially, they were less able to discard the irrelevant feature.
Right. So we made a long tour now from the whisker system into the clinic, and you've been sort of getting us through that every step of the way. And so in that sense, you've been really championing this whole a more system level perspective on the brain and how it relates to behavior. So if you would like to follow in that tradition that you represent, what should be Matthew's law that we have to adhere to?
So I don't have any laws. I don't have any wisdom to pass on to the younger generation. The only
or even
thing
to us,
even to us, I have nothing useful to say. The only thing I could add is that the approach that we use in the laboratory, and I have the approach, nothing works unless you have very, very good students. I have excellent students, very bright and motivated and hardworking, and that's what allows our approach to work. And the approach was I outlined at the very beginning of the talk, which is we construct experiments that have three elements: a controlled sensory stimulus, a behavioral output, and neuronal activity, measurements of neuronal activity. And these three elements we can view as being a triangle, and that gives us three possible connections, the three sides of the triangle. The connection between the stimulus and the behavior gives us some insight into how the brain experiences a sensory stimulus, and this is quantified by psychophysics. We try to study how a sensory stimulus produces a neuronal firing pattern within the sensory system, which we can call coding or sensory coding or encoding, whereby the sensory receptors and then the successive stages of processing put the physical properties of stimulus into the language of the brain, action potentials. Then we want to look at how a sensory representation can lead to a decision, which we can refer to as decision making or decoding or any number of things. So I think that so if you have the right members of your research team, then putting these three elements into an experiment can provide some insights.
But now we want to put this law on a T-shirt, and we don't want to make the print too small.
Yeah.
So what's Matthew's law we can actually print on a T-shirt now?
I think the law is, again, I don't want to call it a law. I'd say something that we have fun doing. Let's call it what we have fun doing instead of the law. I think
it's triangle.
It's the golden triangle, which is I would say is a systems approach. I'd say the approach is that it's more interesting when you actually see the organism doing something. So it's important to have behavior. It's important to know the input, and it's important to know the neuronal basis of this. So to put in a word, I'd say the golden triangle of cognitive neuroscience.
Wouldn't it be better to find a fourth point and call it a diamond?
Well,
Matthew's diamond.
So I can't be the one that does that.
We'll do it for you. Don't you worry. But now, last thing is, look, Tony likes traveling. And so he wants to definitely come and visit you in Trieste 5 years from now, because he's not so quick booking these kinds of things. So, but 5 years from now, he's going to be at your lab to test whether a prediction you made today was actually confirmed or rejected. So what's the key hypothesis that you want to see tested in that time frame?
Well, goodness gracious, that's a tough one. But I think that what I think would be fun, really fun to be be able to do within 5 years is to have rats. We know we could do it in humans. It's not difficult, but to have our animals able to apply two different decision rules to sensory inputs. So we've in the work that I talked about today, we have a comparison task where the stimulus is characterized by intensity and duration, and we can train rats to do an intensity comparison, or other rats we can train to do a time comparison. The stimulus set is the same. So wouldn't it be fun and interesting to be able to train rats in one session or one group of trials to receive the stimuli and make one judgment, and then in the next group of trials to make the other judgment, and to see where in the brain the processing diverges according to what the animal is doing with it. The sensory input would be the same in both groups of trials, but what the brain has to extract to make its choice and its action is different, and it would be intriguing to see where the task rule takes its effect on neuronal processing.
Right. Great. Matthew Diamond, thank you very much for this conversation.
Thank you.
The CSN podcast was produced by the Convergent Science Network of Biomimetics and Biohybrid Systems, a project funded by the European 7th Research Framework Programme. For more interviews, recorded lectures, or upcoming conferences in the field of biomimetics and biohybrid systems, go to csnnetwork.eu. And thank you for listening.